Hello and welcome back to Biology for Beginners. So last week we talked about the molecular structure of DNA, and I know that was probably a bit confusing. Today we're going to talk about DNA replication, and that's not necessarily less confusing, but just hang in there and I know you can get it. As always, about right now you should be seeing the terms and definitions slide popping up somewhere. And you can either pause the video and grab the vocabulary we will be using, or you can pop on over to my Patreon and grab the entirety of my PDF slideshow for absolutely free. However, anyone that signs up to be a patron will get all of the study material as well as this video at least a day early, as well as some other things that I am working on and hope to have prepared within the next couple of months. All right, so let's begin with a little bit of review from last time. So we learned that DNA is a polymer made up of monomers known as nucleotides. The nucleotides are the pyrimidines, which are single-ranged, and they are thymine, which is replaced by uracil in RNA, and cytosine. And then the purines, which are double-ringed, are adenine and guanine. And then the nucleotides have very specific pairing. So A, T, and C, G. Except for if we're talking about RNA, in that case it would be AU. All right, so let's move on to some new information. And let's talk about what's known as the semi-conservative model. And basically this is Watson and Crick's model. You're going to keep hearing about them when we talk about DNA structure because they're the ones that kind of figured it all out. And so what this model basically says is that there's specific pairings of bases in the DNA, and that specific pairing is what makes DNA able to be copied. And you'll get this parental strand of DNA will separate from the other one, and it will act as a template for the new DNA strands to be made. And when you get all finished up with this DNA replication, you will end up with the two daughter molecules that will consist of one parental strand and one new strand. You can kind of see that in this spiral-ish diagram on this slide. The parental DNA molecule and the parental strands are shown in blue, and the daughter strands are shown in that kind of washed out gray color. So kind of looking at that at a more flat diagram that's a little bit less confusing to look at, we begin with this parental DNA molecule, which is all blue, all stitched up together. And then that DNA molecule will separate or kind of unzip, and the new nucleotides will use those parental strands as the kind of template to pop on into place and form that brand new strand of DNA. And then your daughter molecules are made up of one brand new strand of DNA and one parental strand. And that's kind of how it goes. So let's dive into DNA replication itself. And DNA replication is going to begin at these specific areas along the strand of DNA, known as origins of replication. And the origins of replication are just little short stretches of DNA with very specific nucleotide sequences that kind of signal this is where replication is supposed to start. And any single strand of DNA may have many, many origins of replication. And at these origins of replication, the proteins that initiate that DNA replication will attach because they recognize that sequence of nucleotides, and the replication is going to proceed in not one, but two directions, and kind of create what we see here in this diagram is a replication bubble. And as that replication continues, you can see the bubble gets larger, the bubbles will connect to one another, and then in the end, that parental strand ends up opening up and those daughter strands are formed, and you get two daughter DNA molecules. And one thing that's very important about this whole replication process is that DNA strands have what is known as a 5' and a 3' end. And this 5' and 3' are referring actually to the carbons in that sugar molecule that makes up that sugar phosphate backbone. And on that 5' end, as we can see in the diagram, the kind of phosphate group is what's on the end there, and on the 3' end, the OH group is what is on the end. And the 5' end will always be adjacent to the 3' end. So as we see here in this diagram, we have on top, the left side has that 5' end, and on the right it has that 3' end. That's always how it's going to be, and that is very important. But why is that important exactly? Well, it's important because DNA polymerase, which is the molecule that adds nucleotides to that growing daughter strand pretty much continuously, can only add in that 5' to 3' direction. And as it's adding like that, because we're at a bubble, it kind of creates a fork at that replication bubble. But remember, replication is proceeding in both directions. So what's kind of going on here? Well, if we zoom in on this diagram on the little top portion, you'll notice that the one strand is being synthesized pretty much continuously because the daughter strand is running from that 5' to 3' end, which is the direction that DNA polymerase has to run in. That's the only way it can continuously go in. But because replication is always going in 2 directions, so that other strand is being replicated in the opposite direction, it's being synthesized in these little pieces, but DNA polymerase cannot connect those little pieces. So how do they get connected? Well, those little pieces, known as Okazaki fragments, are connected by another kind of thing. They're connected by a molecule known as DNA ligase. And DNA ligase links or ligates the pieces to one another and completes that strand. So kind of the recap here, the big picture here, is that replication begins at origins of replication and proceeds in both directions, which forms a bubble. DNA polymerase makes a continuous strand of DNA from the 5' to 3' end. And if we notice, one of those ends is moving in the kind of wrong direction for DNA polymerase. So it creates the little Okazaki fragments that must then be linked by DNA ligase to create that final daughter strand of DNA. So I know this has been a shorter video, but that's the end of this one today. I know this material can be a little bit confusing, so I don't want to throw too much at you, and the book separates this into its own little section, and I agree with that because that can be quite confusing, especially when we're going back and looking at those diagrams. But that kind of recap slide is the one that has the big deals. They have what you're likely to be tested on. You're going to need to know that replication will begin at those origins of replication. You'll need to know that replication happens in both directions. DNA polymerase makes the new strands of DNA, but on one strand it ends up making it in little pieces that need to be linked by DNA ligase. And you need to know that there's a 5' end and a 3' end, and the 5' end will always be adjacent to that 3' end. Those are the big pictures. That's what you need to take away from this, and that's pretty much what you need to know to get a good grade on the DNA replication section of any quiz or exam in an intro-level biology class. Now, there's more that goes on here if you get into a biochem class, but you probably don't need to know that unless you are going into those more upper div classes, so we're not going to get into that today. But I do hope you have a wonderful rest of your day, and until next time, I will see you all later.
Thank you again for watching this video. Don't forget to like, comment, subscribe, and ring that bell if you would like to see more. And if you'd like to follow me on any of my other social medias, the links are down in the description below. Don't forget to check out thereptilegoth.com for all of my articles and blog posts. If you found any value in this video and you would like to help support the channel, please check out my Patreon page. That link is also in the description down below. And a special thanks goes out to my Diamond Dragon patron, Diane B. What you're doing is really helping me fund a dream here. I will see you guys all in the next one.